So, good evening, participants. Welcome to the day 11 session on applications of bioinformatics. That is majorly on we discuss on systems biology and synthetic biology. In applications of bioinformatics, we have various applications. Some of the applications what we see are other than system biology, synthetic biology. On tomorrow, we'll be discussing on microarray data analysis and DNA computing. And the next day, we see bioinformatics approaches for drug discovery. Related to computer-aided drug design, we discuss on uh, 13th day. And the 14th day session, we discuss on application of bioinformatics in genomics and proteomics. Okay. And finally, the final day, we have name seeking. So this is actually a tentative day for assessment, but you have to confirm with the coordinator. The coordinator will be keeping in touch, and so he will be providing a comfortable date. Okay, but we'll be completing our last session on 29 on Saturday. I mean on Sunday. Okay. So now let me carry out before going to today's session on system biology and synthetic biology. Can we recap the last session topics? So, what are the last session topics we have discussed? Yes, very good. So, once again from Prema Madam, that she has mentioned, we have discussed on protein structure validation. So, actually, protein structure validation can be done through various methods. The most popular method is using a Ramachandran plot to assess the stereochemical quality of a protein. Okay, so even if you see the validation, even the protein refinement resolution. I think I already explained you that even the good quality protein, that is high resolution protein, which is having less than 2 angstrom, then that is the quality. If quality of protein is good, you do whatever you do with computational analysis. You do computational analysis with that protein, you get precise results. Okay, the accuracy would be more when compared to low resolution proteins. And I told you the low resolution proteins, you can find the resolution above 4 angstrom. Okay. So, of course, that is also a kind of validation, like how and what resolution the protein structure is developed. And even if you want to check the protein structure validation, we have other parameters also. The most important Ramachandran plot uses 5 SSI angles in y and x directions. So in that, we can able to find the secondary structure elements of a protein, like where how many alpha helices, beta sheets, loops, turns, and coils are contributing to structure for a protein. If you find, you can also find favorable residues, like that is how many amino acids can favorably form the structure. Then I told you for a good quality structure, we should have the protein quality, that is a number of favored residues in the Ramachandran plot should be 90 percentage and more than 90 percentage. Okay. So through the Ramachandran plot also we can assess through Ramachandran favored regions data present in. So for the Ramachandran plot, I have showed you from Swiss modeler. You remember in Swiss modeler, after prediction of a model, I've gone to the structure assessment. Inside the structure assessment, if you click, it directly takes you to the project tool. Inside the project tool, one can able to see the Ramachandran plot for a proline, lysine, proline, and also we have for overall. So whatever we have for all this, actually generally we see the overall Ramachandran plot to assess the overall quality of a protein. Okay, but we also consider glycine and proline together to find to distinguish the alpha forming and beta forming elements in protein. And if you see the next thing, what we discussed is about how to go for primary structure prediction. That is how to predict physical chemical properties of protein. So we have seen prod parent is one of the tool from XPassive web server. So from the XPassive web server, we can able to find the stability, hydrophobicity, hydrophilicity, number of acidic amino acids, number of basic amino acids, half-life of the protein, okay, hydrophobicity index, molecular formula, molecular weight, etc. And then we have gone for the secondary structure of a protein. For secondary structure of protein, we have gone to the methods like SOPMA, HNN secondary structure prediction, okay, etc. And then we have gone for FIRE. FIRE is for protein homology analogy recognition engine. So actually, this is actually FIRE I have taken for tertiary structure prediction or quaternary. Tertiary or quaternary, we can go with FIRE. So this is also a kind of a homology modeling tool. And then we have discussed about comparative modeling. Comparative modeling, after predicting a computational structure by through any of the methods, homology modeling, threading, ab initio, one has to assess the quality of a protein by comparing the predicted protein structure with the gold standard technique proteins. So then how you can compare? One of the most important formula is RMSD, root mean square deviation. So through the root mean square deviation, one can able to find how much precise your model is predicted from computational method. Okay, and also I told you something about AlphaFold. There is one database called AlphaFold database present in PDB database itself. So if you see the gold standard technique proteins, we have just 2.3 lakh proteins. But AlphaFold, the origin, I think it's not more than 8 years. But you can find around 12 lakh proteins are available just because of the artificial intelligence model used to deposit these structures. And if you see this CASP, see a critical assessment structure prediction, also assess the quality of a protein. So whatever protein structure we predict, we can use a tool called CASP to assess the quality. And also we discuss something about PEPFold. The PEPFold is the server used for predicting the structure for a peptide. What is peptide? The peptide is generally, if you see in immunoinformatics and bioinformatics, mostly the size of the peptide will be 8 amino acid to 50 amino acid. Okay, if you want to design a peptide, you can able to design it in PEPFold. Not only that, even if you want to design a polypeptide, the polypeptide is generally, if the size of the molecule is between 50 to 75, we call that as polypeptide. Okay, 50 to 75 amino acid. And if your molecule is about 75 amino acid, most probably it will be a protein. Okay, so peptide, polypeptide, protein. So for peptide and polypeptide, you can use PEPFold. For the protein structure, can be tertiary or quaternary. We can use the three models, whatever I mentioned, homology modeling. So I also ab initio modeling and threading. And I also shown multiple tools for all these three methods. So one can also practice any of the methods by going to the XPassive observer. So through the XPassive observer, we find all this software. Good. And we also discuss something on PDB sum. So even if we want to predict the binding site, one can go to the PDB sum to find DNA protein interactions, RNA protein interactions, or protein protein interactions. Even in PDB sum, if you have a compound complex with a protein, even if a compound, whatever they've used is a standard drug, okay, so instead of a standard drug, if you're trying with some kind of pharmacognosy, like if you're trying with some kind of natural compounds, you can just remove that compound and you can also check whether your compound is binding in the exactly binding sites of the existing drug or not. So just like a repurposing, you can check the binding sites with the PDB sum through the interaction sites. Okay, so of course, this also we discussed on yesterday. So I think we have come up with all the points. Now let me cover with system biology and synthetic biology. So so many of the institutions are conducting, or creating teams for participation in IGEM, International Genetically Engineered Machines Competition, conducted by Massachusetts Institute of Technology, USA. So whatever IHM, International Genetically Engineered Machines competition, you can find the total application of synthetic biology. Okay, full of synthetic biology is used in that competition. And myself, I am specializing and specialized somewhat in system biology. So in the previous 4 years, so even whatever I got grants, so I'm working on system biology. So for the past 4 years, I'm working on system biology extensively. So okay, now let me come to the topics what we can see in the system biology. So systems biology, if you see, we can I just start from here. So system biology, generally we can say a combination of many subjects like genomics, proteomics, metabolomics, gene networks, transcriptomics, molecular structure. So all these subjects together, okay, we link to study about system biology. Actually, then what is system biology? What is system here? Whether we are going to study any computation system or any biomedical? No, not like that. Here, whatever I'm telling, a system is an organism. It's an organism, it can be an organ. Okay, so generally, uh, if we are studying about human, okay, the human biological system. So if you are taking a drug, how the drug is going to influence on the in the human biological system. Like for example, if you take a specific cancer like a liver cancer, so the most important liver cancer target is of course TP53. TP53, but even this target is important for most of the types of cancer. Okay, for most of the cancer can be ovarian cancer, the colon cancer. You can find TP53 is one of the important target. Then, if a patient is suffering from a liver cancer because of TP53, then whatever drug is going to take, how it's going to exactly target the liver, and how it's not going to target the normal tissues. So that's most important, isn't it? Even though TP53 gene is present in multiple organs, and whatever drug you take, it has to target only the diseased organ. It should not touch the other organs. Okay, so you should understand for this purpose. So you should understand how to target exactly in the disease site, in the cancer site. Okay, I'm taking an example like a cancer, and I'm illustrating you. So for this, one should know about the systems biology. One should know about all these subjects. Okay, if you have these kind of a knowledge, then only we can target specifically an organ and that too specifically a gene, whatever we want to target. Okay, you got it. So system, not only in disease, we are speaking about human. If you are a veterinarian, so you have to, you are speaking with respect to animals. So your major target is to treat animals. And if you are going for a bioprocess, if you are a bioprocess or downstream engineer, downstream processing engineer, one has to know the metabolism of your engineered organism. So one has to know about E. coli, yeast, whatever species you use for culture of, and to produce a specific secondary metabolites. So it is applicable in various areas. So whatever I am telling system biology, not only it is used for just uh, treatment of diseases, can also be used for producing some specific secondary metabolites also. Okay. So in that case, whatever system I speak is the E. coli system. Okay. If you are studying about E. coli. Okay, now let me take you to the system biology approach and how it is integrated with the omic tools. I already told you, system biology is majorly linked with all the types of omics. Genomics, one know that if you study about the genome, we call that as genomics. If we study about RNA, we call it as a transcriptomic study. And if we study about proteins, okay, related proteins, we call that as a proteomics. Then if we study about microbial metabolites, we call that as a metabolomics. We have subclasses also in these all the types of omics. And so system biology is generally linked with all these types of biology. So it links with most of the wet lab techniques. Like here, if you see the genomics, we see uh, next generation sequencing sequencers. And for transcriptoming RNA, we majorly use equipments like microarray, RT-PCR, etc. For protein, One can go with 2D gel electrophoresis, mass spectrometry, etc. And for metabolomics, one can use the HPLC, mass spectrometry, FTR, gas chromatography, etc. So relationship between single and multi-omics data analysis study. So again, a similar kind of how a DNA is linked with RNA, how it can link with the protein. One can know that the DNA is going to transcribe to RNA and RNA is translated to protein. But if you study with all the information of genes, that is, I already told you what is genome. Okay, and if you study about specifically post-translational modifications in the genome, we specifically go for the epigenome study. Okay, the remaining thing we have already discussed just now. Okay, so all these things are important for treatment of any type of cancer. Like if you want to focus on a breast cancer, so you should not just study limit up to the genome and transcriptome. Even if you go to the proteome, whatever drug, whatever gene therapy, or whatever antibody you're going to develop, okay, will be precisely targeting only the breast, not it's going to target with the other organs. If a patient is suffering only with a breast cancer, okay, so one should integrate all the omics. Then only a precise, that is, tailoring of medicine can be done. Okay, precise targeting or tailoring of the medicine to a particular target can be done. So if you see the system biology, uh, we have a language called system biology markup language. In bioinformatics in 2001, they have released this system biology markup language. According to this, okay, we see this, it is a representation format based on XML. You know, XML is extensible markup language. This is actually a database language. To store the data, we use an XML, but they are also integrating this XML. This VML is different, XML is different. Yeah, like if you see even in HTML, okay, you know, HTML also, if you link with, you can also link with XML. XML is mostly linked to almost all the languages. Okay. I think if XML is linked with HTML, you can write a language called XHTML. There is a language called XHTML, you know. Similarly, even the SPML, system biology markup language, can be integrated with the XML for storing the data and replacing the data from XML. So, and if you see the system biology markup language, this represents many different classes of biological phenomena, including metabolic networks. Majorly, we use the system biology markup language to create the networks and to create the cell signaling pathways and to create the regulatory networks. And of course, this will be helpful to get a solution for infectious disease, to get an immediate solution for the up, upcoming infectious diseases. One should use the system biology approach for precise targeting. I told you an example, like if you see Covishield and Covaxin, these are the vaccines generally used to target the initial strain of SARS-CoV-2. So in 2019, whatever initial strain came to existence all over the world, so we can use the basic vaccines like Covishield and Covaxin. But after that, within few months, we can see it's getting rapidly mutated, and almost we got more than 100 mutations, 100 strains of SARS-CoV. If so, one has to focus. Okay, if you are going for a specific strain, one has to go with the SPML. I mean, one has to go with the system biology approach. We have to study what is the difference between the mutated SARS-CoV-2 genome with the existing, with the old SARS-CoV-2 genome. We should find the genes, especially if you're focusing on spike glycoprotein. We have to see what are the amino acids mutated and how about the structure variation. So one has to see how even the structure, and one should also see how the spike glycoprotein is interacting with other proteins. So in that case, protein-protein networks is also important. First, we should know how the structure of spike lipoprotein looks like. The next is you are seeing how the spike is interacting with other proteins, any variations are there or not. So this you have to check with protein-protein networks. And even you can see any cell signaling pathways are going to change. and any kind of regulation of genes are happening or not. So all these things are integrated, You can't just limit up to a single studying about a single task in system biology and system biology markup language. So for this, one can go to this URL, that is spml.org/software. So here you find plenty of tools like lib, SDML, JSDML, etc. So these are some of the programming libraries. We also have format conversion tools, and majorly this URL is, you can able to find tools and programs, etc. Okay, even you can develop our own programs if you know C, C++, C Sharp, Java, JavaScript, MATLAB, Octave, Perl, PHP, personal home page, Python, R and R programming, and Ruby are used. Okay, to develop these kind of formats meant for system biology markup language. So one can also effectively use these kind of programming libraries and you can build your tools. So I can show you some examples from MATLAB, how the system biology markup how we can use the HTML to build the tools. We'll be discussing from MATLAB. So if you see in overall about system biology and integration, I told you here whatever I call as a system is not just an organism. Sometimes the system we can also consider as an organ, or even we can take a cell in an organ, or even organelle, and even up to the molecule. Even a gene and protein can also be considered as a system. So if you see here, in a bacterial biomolecular computing, here I am using a bacterial cell. Okay, I'm taking this bacterial cell as the system. In this system, actually, first of all, you have a software like AutoCAD. You simply drag and put in that kind of a window. You get a cell. Then you have to give some functions like input function, and you have to give some output functions, like how you use a neural network. And it just looks like an AutoCAD. Simply drag and drop the component, and you should feed whatever you want to feed in the input function, and even it can feed something in output function. So if you give any inputs, it gives you an output. For example, if the lactose, if you feed a lactose to the bacterial cell, if it is a pseudomonas type, it can emit the fluorescence. And even the light, whatever it takes, how much light it takes, even it emits that much kind of a fluorescence. And obviously, even based on the temperature, even it can provide an electric current. Even the cells can produce current, isn't it? So the change of pH can also signify you the variation with respect to electric current. So whatever it is, okay, so whatever inputs you provide, okay, you have to write a function, input function, and you have to write an output function, what kind of activation function you can try to get an output. Then after that, you have to feed this. So this is also a kind of a system biology. And here I showed just for as illustrating for a cell, a bacterial cell, how it can use for computing. And if you see similarly, like system biology, you also have something called as network biology. So what is the basic definition for network biology? The network biology is also a branch of science, but it majorly touches these three subjects like biology, network theory. Network theory basically we study the graph theory. In Max, we have something called graph theory. So we study in detail to produce types of networks like gene-gene interaction network, gene-gene regulation networks, protein-protein networks. So we can construct all the types of networks through the graph theory. Okay, RNA protein interaction networks, RNA interaction networks. We have plenty of networks. So with whatever macromolecules we have, they have all the combinations of molecules interactions. Nowadays, we are focusing more on the transcriptomics. So genomics era ended in 2003, 2005 itself. After 2005, the researchers are start developing 1,000 genome, 1 lakh genome, and 10 lakh genomes like this. So actually, after 2005, the proteomics came into existence. Okay, the major area of the researchers was on proteomics. Even at this end at 2015. From 2016 onwards, many people are working on majorly on transcriptomics. Okay. So nowadays they're focusing on other molecules also, like carbohydrate protein interactions, RNA protein interactions, RNA RNA interactions. So many kind of RNA RNA interactions are studying more in this decade. Okay, even though the introns we have like 98% of introns in our body, still they're studying the effect of these introns on production of various proteins from exons. A lot of researchers, whatever is unleashed or being explored at present. Okay. Now, if you see this network biology, we link with the network theory, the form of a graph theory, and we also link a statistics. So the study of these three subjects together, we call this as a network biology. Because if you see even in protein networks, we can see one protein is linked with thousands of proteins in the database. If so, then even you should know. If you have more than 10, more than 100, then obviously one should use the statistics to know about the various functions. We have a p-value generated from protein-protein interaction. What is the p-value? What is the other values? Okay, connected with the interactions. All these things one should know. All these three types of studies. So I told you like even the bioinformatics era, especially we see the network biology, like I told you in 1950 to 1999, we have just complex network theory. So we don't have much computational power, and so we are not aware of networks. We don't know anything about DNA protein networks, protein networks. We have a very poor kind of databases at that time. Only after 99, we have some kind of biological data, and system biology came into existence by the introduction of so many genomes in the genomics, and even proteomes started to develop after getting a final draft of the genomes. Okay, and from 2005 to 2030, contemporary biological applications like models used in real biological contexts, so many ubiquitous complex networks, powerful frameworks. Okay, so these are being worked in network biology. You see networks, as I previously mentioned, we have protein-protein interaction network, also called as PPA, protein DNA, gene correlation, gene regulation, metabolic, and signaling networks. So how a metabolic network looks like. How a protein interaction network. This is a protein interaction network of yeast. You know, 99, more than 99% of the genes code for proteins in yeast. So you find large protein networks in yeast. So this is an example. And here this is a gene regulatory network. You have two different colors. See, one is red color and the other is black color. The red color stands for positive regulation and the black color stands for the negative regulation. So if you take, even it can be in the form of a disease causing gene, you know, this is a TP53. This is a TP53. So there are some kind of proteins interlinked to the TP53. Okay, if for example, I told you in a liver cancer, TP53 is of course one of the important target. But if a patient is diagnosed with TP53 gene, just targeting the TP53 so the drugs alone uh, can't able to reduce the aggravation or can't able to decrease the cancer infection. So one has to, if you want to alleviate the attenuate the, if you want to attenuate the infection, then obviously one has to target the proteins connected with TP53. If you see, there are some nearly 12 proteins are shown here. For example, if you find that IFN, okay, or you find something like TNF, tumor necrosis factor, is a most positive regulating gene, then whatever drug you're going to bind with the TP53, that should also bind with the appropriate binding, that's a TNF. If it is not binding, then you can use some other drug. So along with this drug, if you take a chemotherapy, you can take the second drug for TNF also, because this is one of the interacting protein with TP53. Okay, got it? If you're taking a chemotherapy, then one has to take even the most important gene interacting with TP53. You can take some five positive regulating, upregulating genes and five down-regulating genes. You can also check the binding studies. You can check the other kind of interaction studies, etc. Okay, then whatever you do, whatever drugs you take, and you can able to target the 10 important genes. Definitely, if you give that kind of drugs, then obviously it can give a kind of a remedy for treatment of cancer. That is for tailoring of medicine. Okay, so using system biology approach. So definition of network theory, or how you can understand the graph theory. So if you see this, each and every square, I mean every circle is a kind of a node. You can call this as a node or vertex. Okay. So in our case, if you have a PPI networks, okay. So we have three proteins, 1, 2, and 3. So if you see here, the first protein can also interact with can have an this is a self loop. You see, this is a self loop for the first protein. But if you see the second protein is linked with the first and third through what we call it as edges. So if these are nodes, these are edges. But if you see the third protein, we have three directions. How the first protein is connected with three, this is the shortest path. Whatever I draw an arrow mark is the shortest path. But we also have the other two paths. Okay, so just because we have, if you see which is the shortest path, this is the first shortest path. And the second shortest path is this. So based on this, I am telling. And if you see, you have a white cap. So you have a uh, longest path. So sometimes the interaction can also follows the third path. Okay, how the path is determined only through the graph theory. Okay, like can some proteins can have a short distance in connection, some proteins have a long distance in connected. So one has to find only through the graph theory. So obviously, what you can infer, one can able to find even if one protein is linked with another protein, you may have multi set of edges. And this is a simple graph. When you can tell it is a graph, if and if you have three nodes. If I have just two nodes, and if I just draw a line, what is this? This is a line. Okay, this is a line. This can't be a graph. If only if you have a third node, then only you can have a kind something called a graph. A graph can be considered if and if you have three nodes. So this is a simple graph. Okay, all these things were discussed. I think I can take you to this pathway. This pathway, you can see that glycolysis, gluconeogenesis, citrate cycle, pentose phosphate, all are interlinked with each other. So with this, we can able to find a cake. So I told you, even system biology, we are not just linking with the proteins alone. We also link the metabolites and the pathways. Even the pathways, the network of pathways also we can able to see in system biology. So if you see the most important tools present in system biology, number one, I can tell a visualization of networks that can be done through Cytoscape. So this is the URL to download the Cytoscape. One can install, and after installation, you can use this as an open source software. That is, if you download the protein-protein interaction file, you can open without you can open that networks even without internet. There is also another software called Patchak. Okay, even this is used for visualizing the networks, any types of networks, DNA, protein, anything. And graph related programming, one can also take from basics with LEDA. So this is algorithmsolutions.com. That's the URL for that. One can also go with the Naughty. This is the URL to go with the graph related programming. It is starting from scratch. Okay. So starting from core, if you want to develop, one can go with the graph related programming. Okay. Now synthetic biology. So I just come across with the theoretical concepts, then I can take you to the practical illustration with computational tools. Okay, now I take you to the synthetic biology. Till now, whatever discussed is about the system biology. What is synthetic biology? The synthetic biology is an interdisciplinary branch of biology and engineering. So there you basically study on branch of science, but here you also find an application of engineering. Then what kind of engineering exactly you are saying? Mostly we study about the genetic engineering in synthetic biology. Of course, the genetic engineering biology is linking with a science called molecular biology. and we have molecular engineering. Sometimes we also integrate with the system biology. The synthetic biology also we integrate with system biology. Okay, and one should know about biophysics, electrical engineering, computer engineering. You know why we have to know and I have a knowledge on electrical engineering. So only in electrical engineering we create electrical circuits, PN junction diodes. We have heard about the types of diodes. So you connect various components to form a electrical circuit, isn't it? Similarly, even here, whatever components we have, which can form a gene, so the parts of a gene, we can interlink in the form of a gene circuits. So in electrical engineering, we find the electrical circuits. In our synthetic biology, we find the gene circuits. And of course, one should have a knowledge on computer engineering, control engineering. Okay, so if you have a complex gene circuits, okay, one should know all this information, control engineering and evolutionary biology. Okay, so a lot of science and engineering technologies are involved to know about the synthetic biology. If you see the synthetic biology, this is used to build artificial biological systems. Artificial biological system. You heard something about artificial chromosomes? PACs. We call bacterial artificial chromosome PAC. See, whatever we see a chromosomes from bacteria, now we have the artificial chromosomes for bacteria, yeast, etc. For many species, we have artificial. Not only we have chromosomes, we have so many circuits. Especially, I can take an example like plant biotechnology. Whether you, I think everyone are aware of present GOAs. So earlier in Goa, okay, that is Goa, I mean, jamun fruit, okay, jamun fruit. So if you see this jamun fruit before 15 years, you find very small in size, isn't it? The diameter of guava is very small, and if I have lots, I mean, many seeds inside. So when biting, we find unpleasant with many seeds, and even the how the even the fruit is not that much edible. One should have a strong teeth to bite. But nowadays we have a hybrid guavas. Whatever hybrid guavas we have, it is more crispy. Okay, it's a like a crispy snacks. We are biting the fruit, and we don't find even any seeds. We find pleasant without the seeds. We find even the size is large enough to fill our stomach, and even you find crunchy while eating it. So for getting crunchy, this is they are using a kind of a gene. They are inserting a gene for in the seeds itself for during the production, and for increasing the size, they are imparting a gene, or they are silencing. They are doing a gene silencing with the raw guavas, okay, so that it can grow up to a maximum size. Either they have to go gene silencing or they have to promote the gene, okay. They have to include the gene. So whatever we do that, okay, in to produce the hybrids of guavas, hybrids of tomatoes. Even we can say an example like raw tomato and Bengaluru tomato, hybrid tomatoes. We find plenty of advantages in hybrid tomatoes, like finding it's a red color of a tomato. Only one color you find. You won't find green, orange, etc. You find just a thin skin of the tomato in the hybrid tomatoes, and also you find the size is also somewhat large, and even you find less seeds are present in tomato. So all these things can be used for production of jams, syrups, some kind of ketchups, etc. Isn't it? So what how these are all produced, whatever we do in uh, the genetic engineering to produce the hybrids, everything they initially started with computational techniques like synthetic biology. One day after building the gene circuits, one can easily use this technology to develop this kind of a hybrids. So this is what the application of synthetic biology. And for synthetic biology, if you see, okay, we can use we can develop the synthetic biology circuits okay, through the synthetic biology open language. Like however you find SMO, here you have synthetic biology open language. There you have studied like system biology markup language. Okay, so this is a language we use, and for this is the URL. So one can see all the components of the gene and how to build the circuits using this website. So then what exactly can find in synthetic biology open language? So according to the BioBrick standard, okay, the promoter is generally given by this symbol. And the complementary determining site has this kind of a symbol. Ribosome entry site looks like this, terminator, operator looks like this, insulator. So in the synthetic biology open language, these are the standard symbols used for the types of for the components of the gene, for the components of the gene to use this to build a gene circuit. Okay, for 5' hang we have a different, and 3' overhang we have a different kind of a notations. So one should check through all these symbols before building the circuit. And if you see the synthetic biology is a biological part, is a sequence of DNA that encodes for a biological function. For example, a promoters or protein coding sequences. So one should know what is promoter, what is operator, what is transcript, whatever components we use. Okay, all these things we should know the biological biological application. Okay, and how to integrate. So one after the other, what to link with the next. So all these things one should have in knowledge. So not just a computational biologist can able to use this kind of synthetic biology. Only the biotechnologist, if they have a adequate computer knowledge, then they can sit and they can frame these kind of circuits. Okay. So the basic and component composite parts. A basic part is a functional unit of DNA that cannot be subdivided into smaller components. So here they're giving an example like PBA. This is an example where a promoter regulated by lambda CA is there. Okay, so if you are proficient in genetics, okay, then you can use this information to construct the gene circuits. I think I've already shown in the previous slide. The basic and composite parts. To build the circuits, one has to know about even how to what the genetic engineering, like how a vector can be created from a plasmid, like inserting a DNA in the plasmid. Okay, so first you have to delete the part. Okay, you have to ligate using the plant or specific ends. So we have such a kind of a knowledge. Okay, and one can develop the circuits using the synthetic biology open language. So whatever parts I told promoters. So in the software, you can find in colors, green color. In ribosome binding sites, you find this kind of a color. And protein domains, you find this kind of a symbol and color. Protein coding sequences looks like this. Translational units looks like this. Okay, so in software, you can find all these symbols. So overall, if you see synthetic biology, okay, so whatever we can able to use in genetic engineering to produce hybrids or for other applications, okay, so we can able to work through the software, okay, through the computational prediction. So this is genetic part development. Okay, so here you can able to find the uh, involvement of synthetic biology. Here, a genetic circuit construction, how the genetic circuit is created, so how the regulatory genes and other genes, operator genes are interlinked. So all these things you can able to find here. This functions exactly equivalent to a electrical circuits, like connecting AND gate, OR gate, NOR gate. Okay, so one should know how the AND gate is going to work and OR gate is going to work. So if you understand these principles, you can also know how the gene is going to work. I told you in some cases you have to go for gene silencing. In some cases you have to promote the gene. You have to give uh, so if only if the gene is expressed, then only you can get protein, isn't it? So all these things we have to make it out. Okay, just correlate with the electrical engineering. And also this we can able to test using organoid platform, cut on a chip, and we can measure the efficacy. Okay, so this is something about theoretical part. Now I try to explain using some tools. So if you want to know about the system biology, so one should know about a few basic tools. Remember on earlier sessions, I have taken you to the string DB. Okay, the string DB is the functional protein association networks. If you want to open protein-protein interaction networks, for example, initially I'm going to type TRAP1. TRAP1 is tumor necrosis receptor associated protein 1. This is the heat shock protein 75. For heat shock protein 75, I just type trap1 as the keyword. Now the default it adds to the Homo sapiens, but you also have 261 different species. If you want to see the Homo sapiens, one can click continue. But if you want to see for Drosophila melanogaster, okay, it's just a fruit fly. I'm just clicking continue. Okay, the same HSP75 you can also see in the fruit fly. You know, this HSP75 expresses more when you find a patient is infected from cancer. If you find abnormal tissues are growing in our body, then obviously this expression will be increased to 2.5 to 4 folds. So of course, this is one of the important aspects for cancer. It takes more time. I think the system, the internet is slow. Can I go with the Homo sapiens then? Yes, the Homo sapiens data is fast. Okay, I'm getting like a heat shock protein 75, how it's interlinked with other proteins. So instead of getting these 10 proteins, if you want to get 100 proteins, okay, the top 100 proteins, you can increase to custom value, you can increase to 100. Okay, and if you want to go for more than that, you can also give that value. So now you can see some 100 important proteins. You can find all the colors like, so some proteins are upregulated, some proteins are downregulated. Okay, so you can also find that information. You can also find the information in the analysis. The analysis, you can check all this. Okay, you have the strain false discovery rate and how many proteins are interconnected in the network. Okay, how many nodes are connected? If you see the total nodes, I have a trap one connected with 100 proteins. So total number of nodes are not one, 100 plus one. And the total number of edges, how many lines you can see on connection is 1396. Degree, clustering coefficient. And this is PPI attachment P value. Given how one protein is interacting with another protein, you have a kind of a P value. So this is an interaction. For all the 101 interactions, you have a P value. So expected number of edges, etc. And if you want to download this protein-protein interaction, one can go to the exports and better download the tabulated separated value file. This is the one. So one can download this, or you can use this for if you want to open it in Cytoscape. Okay, download this and open in the Cytoscape. You just download it somewhere in the desktop BIOINFORMATICS demo. Then I don't have anything. I simply download here. So whatever I downloaded here, I just going to open with the Cytoscape. I check whether I installed the Cytoscape or not. Yes, I have Cytoscape. I simply opening the .tsv file in Cytoscape. I had a version like 3.10.1. You can see all these proteins in the form of a network. If you want to have not one protein, so you can first set in the settings, then it can open. If you want just 10 proteins, you go back and just create 10 interactions alone, whatever you present in default. It's asking whether you want to download a new version. No, I don't want a new version. We also have few more sample files to open. Okay, download, no. Yeast gene interactions, yeast perturbation, the colorectal cancer gene. So I'm just opening a kind of a protein-protein interaction. If I have installed the latest Cytoscape, okay, if I go to the network, here you have send network to Cytoscape. There is an option in our website to click send network to a Cytoscape. If you click that, directly it opens a network. Okay. Just because we have 100 proteins in network, it takes some time to open. Okay. So meanwhile, I can go to the, it takes some time to open. If it opens, then I can show you. So meanwhile, I can go to the next website. Like a string, one can also see the networks of the fun book database. And if you want to have such a kind of DNA protein networks, RNA protein network, you can see some private databases. We can also have interactomic databases, transcriptome databases, etc. So this is a kind of, again, a protein database. Okay. So you can type one kind of protein, you can search or spike glycoprotein, or even you can type like spike and you can search for it. Now I'm simply typing the trap and searching this. Okay, so for all the networks, we can use this kind of websites. So if you want to see the specific gene protein networks, one can even go to the UniProt. In UniProt, we have plenty of hyperlinks to interconnect with types of networks. So for example, here I'm going to give an insulin. Okay, if you want to see what are the types of networks present in UniProt, you just go to the human insulin, go to a specific protein. I'm taking to the human insulin ligand. Interactions. Okay, see here. What are the types of interactions? For protein, you have BioGrid, database of interacting proteins, DIP, intact, mint. I just showed you the string alone, isn't it? This is the string identifier. Other than this, we also have plenty of databases. This is also a largest database. See, 525 interactors are there. When compared to other databases, here also you can find more interactions in BioGrid. Okay, and even RNA Act. That is how the RNA protein interactions you can find here. RNA Act. Okay, so likewise, okay, in few proteins we have also DNA protein interactions and other kind of interactions also. Okay, other molecule protein interactions. So this is an RNA Act. So this is gives you an information of how RNA is interacting with that specific protein. Okay, one can find more information from this. Okay, and this is the server for protein RNA interactions predictions. So one has to type a specific RNA binding protein. RNA binding protein, you can take RNA dependent RNA polymerase. Even RNA dependent RNA polymerase is most important target for RNA viruses. So one can type RNA dependent RNA polymerase, you can search, and if you can go to the specific virus, and you can also find that. Okay. RNA dependent. Whatever. Or we can give simply the examples, whatever we find at the bottom. No, I didn't give polymerase. So meanwhile, I open the cytoscape. It's still running. I might have downloaded 10 proteins, 10 network protein. Then it might have opened. Okay, I reduce the string and the string instead of 100, I'm going to give 10. And simply after clicking 10, update. So I got this in exports. I go to the exports. I download this Excel cytoscape. Okay, download. Now I'm downloading this as short underscore 2 dot tsv. Of course, this is a tsv file. I downloaded. Now, whatever I downloaded, I try to open once again in the cytoscape. Either you can open like that or simply drag and drop. Drag and drop. File new session. Still loading. I think if I've updated the next cytoscape, there won't be any problem. Immediately get a kind of no. So I'm just opening from the option present here. So this is the network. Okay. So I just click here how to import a file. So from here, I just imported the thing. Here I can find the information of how the proteins are interconnected in the network. So instead of showing this data from, if you take this data and put in the publication, you get in a problem with plagiarism. So one can try with this. You can change the customize, you can change the colors, you can do whatever you want, and we can able to use this image for publications. So one has to try to use this cytoscape for all the types of networks interactions. Okay, this is a network from Funko. From the trap one, so you're getting more significant proteins. One can also use the advanced filters. If I'm focusing on breast cancer, I'm clicking a breast. And then if you're focusing on a specific pathway like PA3K pathway, so only one protein is involved. Okay. So only one protein is this is the most important target because others are hidden. Okay, other nodes are hidden. So this is the most important target. So one has to click here and get the details of the protein and it can target accordingly. Okay. So this is from Funko. The RNA interaction still it's getting loaded. I think better I can use some example. TDP43. Okay. After some time we can find the RNA protein interactions. Okay, now I also can take you to the next software that is MATLAB. This is also a kind of system biology software. So MATLAB, I've installed 2024 version. Now presently we also have 2023 version. You know why I'm just opening MATLAB for system biology? We have a module, we have a program to work with system biology in MATLAB. So this is the most widely used package for practicing the system biology application. Yes, MATLAB is getting loaded. So meanwhile, I can open the RNA protein, not yet loaded. So I can close the other windows, make it faster. Okay, meanwhile, I can see the MATLAB is open. So here I show you the components present in for system biology, and I tell you how to practice. You don't know anything about MATLAB, how to learn the MATLAB, and how to work with it. So if you see here, uh, in MATLAB, this is the window of a MATLAB, where you can also practice MATLAB through the MATLAB commands. Okay, if you want to learn the MATLAB, okay. So if you're the beginner, if you click here, you can find some kind of a tutorials, online tutorials. It gives step by step. Like you have different kind of courses, like how you find course in Coursera and edX. There are so many kind of platforms, isn't it, to study about the courses like this. Here also we have courses for MATLAB. One can, if you have a license of MATLAB, one can also go and learn, and you can also secure a kind of a certificates from the MATLAB. Okay. So, and if you see here, what are the other options present? If you want to work with system biology, I'm clicking apps. You have Thin Biology Model Builder. You have Thin Biology Model Analyzer. Okay, and if you want to know how to use this, you have help. In help, you have documentation. So in home, help, you have documentation. If you want to practice with example, one can specifically select SimBiology example. Okay, so documentation, if you click this documentation, take you to the documentation. Uh, so, but you should connect with the internet. Okay, if you want to work with a specific SimBiology tool, you can go to the specific SimBiology and you can learn. If you want to use other options, okay, other applications, if you want to work with computational biology, click here and try to learn. For example, if you want to see what's there in computational biology, one can see here, SimBiology is there in the computational biology. Even bioinformatics toolbox is there. So you have to click SynBiology inside. Okay, even for next generation sequencing, you have much of the information. One can download even the PDF documentation. So what are the SynBiology apps present? Modeling simulation. So all this information you can find from the help center. So how I gone to the help, home, help, and then I click the documentation. For example, so click here. So now I'm just taking you to the apps. Okay, I'm clicking the system biology model filter. So now a window is open for that. So here you have to open a file. Now I just already saved a file called a project file for a tumor growth model. So I simply open this tumor growth model. This is a sample file. I'm opening from the system biology builder app. So now we're going to get a kind of a multi-components. So see, actually you find if you know something about pharmacokinetics, we have compartment models, central compartment, peripheral compartment. So this is a tumor growth model. One can feed the values. One can feed the dose of the drug, whatever it's going to give for an animal. And if this is for an animal model, so you can feed those values and you can check how much drug is utilized in a particular central compartment. Like in the central compartment, if you're targeting liver cancer, how much a liver is going to how much drug is taken in the liver and how much drug is taken in the blood it can take in the form of a peripheral, and we can also study about the growth model, etc. Okay, so whatever we study with the animals or humans, we can simulate here using this symbology tool present in MATLAB. So there is an option for us called model analyzer. I see after opening that model, I click this uh, model analyzer, click run. Now you are going to get a kind of a curve. Like for the values whatever fed in default in the sample, okay, how you can able to get to actually they have given like paclitaxel. This is a drug used for treatment of cancer, for breast cancer. So if 30 milligram of paclitaxel is given, okay, after 8 days, how you can find the efficacy? So actually, not only that, they have tried for 40 days. For 40 days, the utilization of a drug, okay, the central compartments, peripheral compartments, everything they have shown something like a bioavailability, bioequivalence. These charts can be analyzed using the system biology tool and system biology analyzer tool. Okay. So we can also give the any other drugs and we can also give the data. Even if you feed the data, then only can get these kind of results if you to go for analyzing. So there is a lot to learn. I just completing with the basics. Okay, but one has to have a license and he has to sit to work on these to find uh, a significance of system biology. Okay, so I have completed my session at present. Do you have any doubts at present? No, MATLAB is a commercial version. But our institution, Sri Venkateshwara College of Engineering, Sri Paramudur, has a license version, and each and every student are afforded with a license. So the one license for a year, I think it's around 69,000 approximately. I'm not confident on that, but each and every faculty and student has an individual license. So either we can download and install and work with an offline, or even we have a MATLAB online software. If you have a license and if your system is not having any adequate space, you can even work with the MATLAB online software. It's a commercial software. If you have a crack version, you can try to use that also. Okay. Any other doubts you have at present? If no doubts, okay. Thank you for your interaction. Okay. So, so on next session, okay, we discuss on microarray data analysis and DNA computing. Thank you.